gets lunch. I'm going to just jump in, Sandy, unless you want to do it, and introduce Jim Bowen from the University of Washington, who's here to tell us about multiple sclerosis.
Well, even though Seattle's the rainy city, it has rained only one day since June. And I'm getting kind of dried out, so I'd like to thank the conference organizers for greeting me with that huge downpour at the airport last night. I'm fully hydrated, and my dry sense of humor is a little less dry now. Well, I'm supposed to cover all of multiple sclerosis. I have hundreds of slides and many hours of lectures, but people are already shaking their head no. So I thought I better cut it down to just a few important points. So I tried to limit it and just talk about some of the new things in the field. One of them is how to diagnose it, also using that, how to determine what the risk of going on to get MS is after the initial presentation. Some new data indicating why we should recognize this as early as possible and start treating it. And then finally, some of the recent pathology developments. So first, the diagnosis. The diagnosis hinges on the multiple part of multiple sclerosis. You need multiple areas in space within the nervous system and multiple in time. The old criteria were the Poser criteria and were developed in the 70s. And you've seen all these pictures of MRI scans today. Well, MRI came out in 1985, so the criteria that we were working off of did not even, MRI didn't even exist. So the new McDonald criteria incorporate MRI into the definition now. So these criteria require at least 2 attacks at different times, at least 2 locations in the nervous system, and then the really difficult one, no other explanation. So if you look at the attacks in time, if a person has only 1 attack, well, you need the other one. So your choice is that you can just wait until the other shoe drops, or you can start doing MRIs. And they've defined how this is done. Everything within the first 3-month period is considered part of the same attack, so you have to get your baseline MRI at 3 months and then do serial MRIs afterwards. So a lot of you that presented with transverse myelitis will find that your doctors are doing serial MRIs trying to catch a second attack in time. The evidence is that for every attack an MS patient is aware of, there are approximately 10 silent ones on MRI, so that it's got a fairly high yield to do this way. If a person has only 1 location, transverse myelitis, you don't have MS yet. You have to wait until they have a second location. So your choices are that you can just wait until they have another one, or again, you can use the MRI. It was hard to figure out how much MRI change you need to have to qualify for MS. There's one particularly good study on it by this German named Barkhof, and in a true good German form, he did an experiment and picked the result that was absolutely the most scientifically valid, and here it is. So you need 9 lesions. I think an American would round it off to 10, but 9 was the number that worked best in the models. So you need 3 out of these 4, either 9 lesions or 1 enhancing lesion, 1 below the tentorium, 1 that's just below the cortex, or 3 or more periventricular ones. We don't know what to do with spinal cord lesions. That wasn't included in the model, so it was arbitrarily decided you could substitute 1 cord lesion for 1 brain lesion and get away with 8. So that's the 2 major criteria. The other problem with the old criteria were that some patients have primary progressive MS where they never ever have a single attack. So you could never meet the criteria for multiple episodes in time because they never even had the first one. They just have a slowly worsening spinal cord presentation. So the McDonald Criteria provides for primary progressive MS diagnosis. It requires that spinal fluid be abnormal. This is probably going to be an issue as time passes because we have pretty reasonable data from the PRISMS study, which is a primary progressive study, that a third or so of people with primary progressive MS have normal spinal fluid. So we're going to miss those. They also had to progress over a year, and you can do that by either clinically following them or looking at MRI. And then they have to have some combination of these MRI findings to qualify for the multiple episodes in space. It's not working. There we go. So you can see anytime it takes 3 slides for me to just give a brief overview of the diagnostic criteria, you can see why your docs are struggling so much with getting the diagnosis right. It's very hard to do. The average MS patient takes about 3.5 years to reach a diagnosis from the time they got their first symptom. Well, how do these criteria perform relative to the old ones? This is rather complicated, but start with a box down here. This was 139 patients with CIS, clinically isolated syndrome. This means they had one attack of something, like transverse myelitis, optic neuritis. And they, at 12 months, got an MRI. And within that 12 months, 37% of them had a new lesion and therefore met the McDonald criteria for multiple episodes in time. Within the first year on the old criteria, only 11% of them had met that criteria by having a clinical attack. They went on and they took these people that met the McDonald criteria at a year and saw how they did. And the dark bars are that by the 3rd year, 80% of them had gone on to have a clinical attack. So it seemed the MRI predicted quite well who was going to go on and have a more aggressive course. The light bars are ones that had no change on the MRI at a year, and you can see only 20% of them had gone on to get MS. So if you compare this 80% 3-year rate to the Poser criteria where only 44% had gotten it, we're getting almost twice the yield with these new diagnostic criteria. Well, how do we use these to determine a person's risk of whether they have MS and whether they should be treated? There's one good study with this that has generated a huge number of papers. This was the 1993 5-year paper, just to show you that those that presented with transverse myelitis, if the MRI was abnormal, 2/3 of those had gone on to get MS within the 5 years. This has now been updated with 14-year follow-up on this group. And if you look at the line that's in bold, if they had even between 1 and 3 spots on their MRI 14 years ago when they first presented, and this is a brain MRI, even though they might have transverse myelitis, 89% of those had CDMS, that's clinically definite MS, and another 6% had clinically probable. So you're getting them at 95% chance that they have MS. So because of this, if a person presents with a single episode like transverse myelitis and their brain MRI is abnormal, most MS specialists would recommend going ahead and treating those people, knowing that their risk of really having MS is extremely high. Well, why treat it early? Why not just wait for the next attack? Well, there are several lines of evidence suggesting that we should treat this early, and I'll go over each of these in turn. Here's evidence from atrophy, changes in normal appearing white matter of the brain, functional MRI changes, and then data from 3 drug trials. So let me go over each of those. Here's the atrophy data. There are many, many reports of this, but I thought Jack Simon's work showed this pretty well. These are 4 different measures of atrophy on the brain, or shrinkage, and it doesn't really matter which one you pick. But they took people with an EDSS of between 1 and 3.5. So EDSS is a disability rating scale, and 1 to 3.5 is the lowest end of the scale. So 3.5, they're not really even having trouble walking at that point. So very, very mild MS. And then they followed them, and the white bars are the changes in atrophy at 1 year, and the dark bars are what happened by 2 years. These two on the right, when it goes down, that's bad, and the first two on the left, when it goes up, that's bad. So basically, bad things happen to the atrophy, no matter how you looked at it, and that this was determined very early in disease, and even at 1 and 2 years afterwards. So atrophy is occurring, and we'd like to jump in there early with treatment to try to prevent this. This is MR spectroscopy. This is a picture from our scanner at the University of Washington, and you've seen all these regular pictures of MRIs that the earlier speakers have shown, and to be honest, those are kind of old and getting boring nowadays. So we have this new fancier technique, and what we can do is, you can see the little white box, we can actually set the machine not only to get a picture of the anatomy, but to get this chemical footprint of what's happening within that box. This NAA peak is a chemical that is in axons and is a measure of axon integrity. The choline is in myelin, so we can actually measure how much damage there is on a chemical basis within that square. So in this study by Fernando, they took clinically isolated syndrome. Again, this is people that have had a single attack, and they looked not at the spots on the MRI, but at the normal appearing white matter. So totally normal looking areas of the brain. And they found that looking at these various chemicals, they're pretty similar between a control group and the people that had a single attack, until you get down to the myo-inositol. And that is elevated in those with the attack. This chemical is a measure of the metabolism of oligodendrocytes. So it looks like very early on, right after the first attack, even in the normal areas of the brain, the oligodendrocytes are already suffering. And we should, again, jump in early and try to prevent them from dying off. This is the functional MRI, and with this, this is again from our machine. We're able to have the person just lie in the machine, get a regular MRI scan, and then we have them do something. In this case, they were tapping the right index finger like this. We have them do that for 10 or 30 seconds, and then we alternate between lying there doing nothing and doing it for 30 seconds. And we can actually measure the difference in blood, in oxygen extraction from the blood with them doing this. So these lighted up areas on here are areas of the brain that it required for this person to tap their finger. Well, as you probably remember from biology way back, the brain is cross-wired so that the left brain does the right index finger. What you probably didn't know is that MRIs are cross-presented so that the left side of the brain is the one on your right, and the audiences are cross-presented. It's all very confusing. But the bottom line is that this spot right here is the one that ought to be lit up to tap the right index finger. And you can see this person has widespread changes across a big area of the brain. This person has MS, but they have it so mild that you would not be able to tell it seeing them walk down the street. They're very mildly affected, but yet at this very early stage, they're already having to use considerable more brain resources to do simple movements than a normal person. This is another study by Barkhof. He looked at clinically isolated syndromes again, so these are people that have had their first attack. And he found that these three areas of the brain that with functional MRI, they were lighting up, and they shouldn't be. So again, very early on, there are wider spread changes in the brain than the regular MRI would lead you to believe. There's evidence from the drug treatment trials that treating it early works and is probably a good thing for the patients. The Copaxone study, the patients were treated for 2 years on the study. Half of them were given the Copaxone, the other half placebo, and at the end of 2 years, that was the end of the study, and they put everybody on drug then. The placebo group crossed over to be on drug, and they followed them now for 8 years. And over that 8 years, the number of relapses that they had progressively fell. It was about 1.5 attacks per year at baseline, and by 8 years, it was 0.16. We don't know if that is because the drug just gets better and better with time. It could be that, or it could be, we do know that patients have fewer attacks with time, or it could be that the ones that were having attacks dropped out of the study, so you have only the good ones left. It's hard to interpret that, but what is easier to interpret is if you look down here, 35% of the group that had been on Copaxone for the entire 8 years had had a worsening by 1 point on a disability scale. But if you look at the group that was on placebo for 2 years and then on the drug for 6, 50% of those had gotten worse. So this 15% difference is what happens when you don't treat people for the first 2 years. What ground you lose during that first 2 years, you never make up. This is the CHAMPS study. For those who follow MS things, you got to have a cool name for your study. So here's what it all stands for. This was a AVONEX study, and they took people with optic neuritis, brainstem, or transverse myelitis presentations, and half of them got AVONEX, the other half got placebo. And these were people after their first attack, and you can see that the AVONEX group did better than the placebo group. So we know this drug works, even given at the time of their first attack. This is the ETOM study, which is another big, long name that supports the use of ETOMs, but this one is Rebif, and again, it was Rebif versus placebo. And you can see that there was a difference in the development of second attacks. So this drug works early on. This is the PRISMS long-term follow-up. This one is, again, with Rebif. For that one, they put everybody, half the patients on placebo, half on Rebif for 2 years, and then they crossed everybody over to drug, similar to the Copaxone design. And you can see that these, the blue and the green bar are how many attacks people were having that were treated for the entire length of the study. The red bar is what happens if you don't treat them the first 2 years and then switch them over. They have a higher relapse rate. So again, what you lose in the first 2 years, you don't gain back by treating them later. So those are the reasons why there's an emphasis in trying to recognize MS early and get on with treatment. The last section of the talk, I'd like to address briefly some of the pathology changes, and some of the other speakers have alluded to that, but I think that this is really revolutionizing our way of thinking about MS. We used to think it was one disease, but now it turns out there are 4 pathology types. This work was done by Claudia Lucanetti at Mayo, but Hans Lassmann, who is the speaker on the scientific half, was the European counterpart that participated in these studies. So there are 4 types, and this is type 2. Type 1 and type 2 look the same, which is why there is no type 1 slide. But you have the T lymphocytes and macrophages that people talked about. This big white spot here is missing myelin with a stain for myelin. On this right-hand panel, the dark spot is a macrophage stain, so the area that's missing the myelin is just chock full of macrophages. Macrophages are the garbage disposal units of the immune system, so they're in there cleaning up debris. Down here on the bottom is a measure of complement. Antibodies and complement often go together to cause damage. So the difference in type 1 and type 2 is that type 1 does not have complement or antibodies, and type 2 does. There's nothing really that new about these findings, and this is pretty much what we expected to find. Type 3, however, was a surprise. Again, on this one, the right-hand panel is the missing myelin, and you can see the macrophages involved there. The borders of this one are not as crisp as the previous one because the line of inflammation is not as well demarcated. Down on the bottom, the interesting thing is that these are pictures of dying oligodendrocytes, and you can see in the middle their nucleus is black. This is stained with a tunnel stain for the scientist here. This is something called apoptosis, and apoptosis is like a suicide pill that we're pre-programmed to have. We use it a lot, like cells that are infected by viruses will trigger apoptosis and kill themselves off rather than bringing down your entire body. During formation of the fetus, we grow 10 times as many nerve cells as we need and kill off 90% of them by apoptosis, leaving us hopefully with the best 10%, though that's clearly not the case in all circles. So these cells are dying, and they're dying by killing themselves. They also do not die right, well, they do die right within the lesion, but they also die out from the lesion a ways in areas that there is no inflammation going on. And then they also have a preferential loss of MAG, which is a protein within the myelin, but it's on the inside of the myelin. So they're dying from the inside out. Whereas if it were an immune system attack on the myelin, they would die from the outside in. So these findings are curious. Type 4 is also curious. Again, here's the lesion on the bottom, and there's 3 good pictures of it. And if you look at these lesions out from the lesion, there are these white holes out a ways. Those are holes left by dying oligodendrocytes. So again, the oligodendrocytes are dying away from the lesion, out from the wavefront of it. These are just dying a regular death. They're not dying through apoptosis. So what do we, why is this a big deal? Well, it's interesting that there are 4 types. So now we don't know, is MS one disease that has 4 stages, or is it 4 diseases that are all masquerading as the same? We don't know. Type 3 and type 4 have never been associated with autoimmune processes. So all the talk about autoimmunity, it makes you question, well, does that apply to type 3 and type 4? It's clear the immune system is up to no good even in those types because it's involved in cleaning up the debris and stuff, and you can get collateral damage from that, but it's made us rethink our approach to the whole disease. Type 3 and 4 have only been seen in virus infections, toxins, or diseases of where the oligodendrocytes get ill of their own accord. So we don't know which of these illnesses are contributing to type 3 and 4. Type 3 and 4, by the way, make up probably 30 or 35%, somewhere in that range of the MS realm. Each patient only has one type. Now, these pathology studies were mostly autopsies, and of course, patients only have one of those. But there were a few patients that had a biopsy, and then either another biopsy or another or an autopsy. So the few cases we have that we have more than one time point, they seem to maintain the same pathology, which suggests that maybe it's 4 diseases masquerading as one. So with that, I'll hopefully it's given you something to think about. I hope I've been able to tell you a bit about the McDonald Criteria and how we're now able to diagnose MS earlier and more accurately than before. Hopefully you understand why we have such an emphasis on MS in a conference that is heavy on transverse myelitis, because it's so important for us to separate MS out from that so that we can treat it early to prevent the changes I was telling you about. And then finally, understand that we don't know what causes MS, but that the recent findings from pathology have led us to really rethink what we know about this. So, thanks.